Bismillah ar-Rahman ar-Rahim. A point mutation may be defined as an alteration in nucleotide sequence, whereas in frameshift mutations the reading frame is altered. Mutations that result from damage to the nucleotides of DNA molecule or from unrepaired errors during replication can be transcribed into messenger RNA and therefore can result in the translation of a protein with an abnormal amino acid sequence. When gene sequences are given, they refer to the coding strand. A gene consists of the transcribed region plus the regions that regulate transcription of the gene like promoters and enhancers. Point mutations are caused by change in a single nucleotide. In case of silent mutation, the codon containing the changed base may code for the same amino acid. For example, if the serine codon UCA is changed at the third base and becomes UCU, it still codes for serine. This is termed a silent mutation. Silent mutations can lead to disease if they create a new splice site during RNA processing. Hutchison-Gilford progeria syndrome, a premature aging disorder, can result from a mutation in lamin A gene, in which base at position 1824 in the gene is altered from a cytosine to a thymine. This single nucleotide change does not alter the amino acid sequence, as the altered codon still codes for glycine, but it does create a cryptic splice site in exon 2, which gives rise to pre-laminin A, that is missing 50 amino acids. The loss of these 50 amino acids interferes with post-translational processing of the protein, as well as the disruption of nuclear membrane, telomere dysfunction, chromatin remodeling defects, and epigenetic alterations. In missense point mutations, one amino acid in a protein is replaced by another amino acid. The codon containing the changed base may code for a different amino acid. For example, if serine codon UCA is changed at first base and becomes CCA, it will code for a different amino acid, proline. This is termed missense mutation. So here you can see that the codon now codes for proline instead of serine. Sickle cell anemia is caused by a missense mutation in each of the alleles for the beta globin. DNA has a single base change. In sickle cell gene, GTG replaces normal GAG. Thus, in the messenger RNA, the codon GUG replaces GAG and a valine residue replaces a glutamate residue at position 6 in the protein. A nonsense mutation causes premature termination of a polypeptide chain. In case of nonsense mutation, the codon containing the changed base may become a termination codon. For example, if the serine codon UCA is changed at its second base and becomes UAA, the new codon causes premature termination of translation at that point and production of a shortened truncated protein. This is termed a nonsense mutation. One type of thalassemia is caused by nonsense mutation. Codon 16 of the beta globin chain is changed from UGG to UGA. This results in conversion of a codon for tryptophan residue to a stop codon. Trinucleotide repeat expansions may occur in the coding or non-coding regions. Occasionally, sequences of 3 bases that are repeated in tandem, which means that are repeated one after another, may become amplified, and too many copies of triplet may occur. Now, if this occurs in the coding region of a gene, the protein will contain many extra copies of one amino acid. For example, expansion of CAG codon in exon 1 of the gene for Huntington protein leads to insertion of many extra glutamate residues in the protein, causing neurodegenerative disorder. These additional glutamates result in abnormally long protein that is cleaved, producing toxic fragments that aggregate in neurons. Sometimes in trinucleotide repeat expansion, a codon may become amplified in the non-coding region. If trinucleotide repeat expansion occurs in an untranslated region of a gene, the result can be decrease in the amount of protein produced, as seen in Fragile X syndrome and myotonic dystrophy. In Fragile X syndrome, which is the most common cause of intellectual disability in males, the expansion results in gene silencing through DNA hypermethylation. Mutation at splice sites can alter the way in which introns are removed from free messenger RNA molecules, producing aberrant proteins. In myotonic dystrophy, gene silencing is the result of splicing alterations due to triplet expansion. A frameshift mutation occurs when the number of inserted or deleted nucleotides is not a multiple of 3. If one or two nucleotides are either deleted or added to the coding region of the messenger RNA, a frameshift mutation occurs, altering the reading frame. This can result in a protein with radically different amino acid sequence or truncated protein due to eventual creation of a stop codon. If 3 nucleotides are added, a new amino acid is added to the protein. If 3 nucleotides are deleted, an amino acid is lost. Loss of 3 nucleotides maintains the reading frame but can result in serious pathology. So, point mutations may be silent mutations, missense mutations, and nonsense mutations, whereas frameshift mutations result from loss of one or two nucleotides, and then there may be trinucleotide repeat expansions which may occur in coding or the non coding region.